Welcome to the presentation Dancing in the Dark, the never-resting ballet of animal life under the Arctic sea ice. This presentation gives a peek view on the faunistic work during the MOSAiC expedition and a brief outlook which scientific directions we might take in the elaboration of our results. The metadata and preliminary results shown in this presentation are truly a mosaic of a work of many people within the ecosystems team of the MOSAiC expedition. The Arctic Ocean is at the front line of the climate crisis. Its thick sea ice cover is declining rapidly. We see a new Arctic emerging, changing from perennial to seasonal ice coverage with thinner and more dynamic ice flows. In addition, warm and salty Atlantic water intrudes further and further into the Arctic basin. It brings not only heat, but also biological communities from lower latitudes. These changes increase pressure on the endemic cold-adapted and sea ice-associated or sympagic wildlife. We are seeing first declines in species numbers and abundances of sympagic fauna, and models predict increasing pressure on iconic species, such as the polar cod. The new Arctic brings new species, such as boreal fishes and Atlantic predators. They are both predators and competitors of the resident Arctic fauna and foreshadows of the new Arctic ecosystem. These changes will profoundly transform ecosystems not only on the shelves but also in the ice-covered basin. Particularly in the deep central Arctic Ocean, our ability to understand the impact of this ecological transformation is limited because of large knowledge gaps regarding pelagic and sympagic animal life and especially their seasonal dynamics. The MOSAiC expedition offered the unique opportunity to close many of these knowledge gaps. The science during MOSAiC had a strictly holistic approach, combining multidisciplinary research in the atmosphere, sea ice, ocean, and ecosystem. During MOSAiC, the German research vessel Polarstern followed the transpolar drift from start to end, covering a distance of 4,300 kilometers. The scientific activities encompassed 312 days. Altogether, more than 700 scientists on board and on land have been working together in this expedition. The objective of the faunistic work was to investigate the seasonal variability of animal diversity, biomass and production, and their contribution to biogeochemical cycles. To this end, we used a variety of sampling gear, including vertical nets, the imaging profiler Loki, the under-ice sampler ROVnet, video observations, echo sounders, and ice corers. In the water column, we used many nets, but also modern sensor technology. We used 6 different types of nets, sampling down to 2,000 meters water depth. And here we can see how the sampling was distributed across the Arctic basin. This means that we could well document the seasonal and geographic change of the dynamics of the animal community. The diversity of life we encountered was astonishing, ranging from unicellular organisms to fishes, an impressive documentation of the tree of life in the Arctic Ocean. This collection of samples will constitute a reference to the continuing change of the Arctic ecosystem. Some preliminary data show both well-known and not so well-known processes. Here we see how the descent of the dominant copepod Calanus hyperboreus during freeze-up goes together with reduced feeding as soon as lipid reserves are filled. Data from the imaging profiler Loki show that reproduction started even very early in the winter season, not only in copepods but also in the ice amphipod Apharusa glacialis. Loki was mostly deployed in parallel with the nets, proving full coverage of the drift. Here we see 2 examples of Loki profiles. The y-axis shows the depth, and the x-axis shows the number of pictures taken per 10 meters in the left panel, and then temperature, salinity, fluorescence, and oxygen saturation. The number of pictures is a proxy of zooplankton abundance, but the data need more processing to become true abundances and show the species distribution. The top row indicates a concentration of animals very close to the surface at the onset of winter 2019. The bottom row shows a deep maximum at about 300-meter depth in late summer 2020. These are typical profiles of seasonal changes in depth distribution. Such distribution shifts were also observed in the multi-frequency echo sounder on Polarstern. With MOSAiC, we were able to conduct the first trans-Arctic hydroacoustic survey. In fact, including transits, the survey comprised 3 crossings of the Arctic Basin in the end. In total, we covered about 9,000 kilometers during 350 days of observations. These data will be analyzed in an EU project investigating the potential distribution of pelagic fish and their prey. If you are interested in more unique hydroacoustic data from the Central Arctic Ocean, have a look at the booth of Giulia Castellani. In the sea ice, we used a variety of techniques to sample this challenging habitat. For ice-in fauna, we took ice cores. Under ice fauna and subsurface zooplankton were sampled with a newly developed net mounted on an ROV. And we had to be quite creative to sample organisms in this habitat. The ROV net scrapes off animals from the sea ice bottom, but it also samples pelagic animals. Standard depths were under ice and 10 meters, but we also went deeper to target specific layers of zooplankton. But some animals were simply hard to catch with these methods. For example, these polar cod, hiding in a crevice. So my colleagues Serdar Sakinan and Giulia Castellani finally used their bare hands many times, securing most precious specimens for our analysis. This video shows footage from a camera mounted on the ROV and recording every single dive. It shows a few examples of observations of polar cod at the underside of sea ice. Aren't they adorable? However, someone else is interested in them too. Now we know why they are hiding so well. On the left side, we see the number of observations of polar cod per minute and their geographic position from November 2019 to the beginning of March 2020. They basically show that polar cod were practically always present. On the right, we see a quick and dirty sketch of their depth distribution. The apparent downward shift is probably related to a greater depth of ridges during the second half of the observation period. Further analysis of the full dataset from November 2019 to September 2020 could show us if the association of polar cod with the sea ice changed during the drift and which conditions trigger distribution changes. But there was much more to see under the ice. Other predators, such as jellyfish and arrowworms, were always present too. If you want to learn more about the beautiful jellyfish, drop by at the booth of Charlotte Havermans. What do we take home from this? With MOSAiC, we were able to study the biodiversity, distribution, life cycles, and physiological dynamics of Arctic animals for a full annual cycle. And here we see the distribution of different samples. Altogether, this endeavor yielded more than 9,000 samples of about 20 parameters for the many analyses we have started to perform. But data analysis has only just begun. So far, based on these first observations, one working hypothesis could be that there is much more life in the Central Arctic Ocean than we thought before, because A, there could be more biomass that could not be detected by past methods due to limitations in spatial and temporal resolution and size of animals targeted. B, animals are active year-round, requiring continuous energy supply, and this means prey, and thus more life. There are many predators, such as jellyfish and polar cod, indicating significant prey stocks to sustain them. Based on the MOSAiC experience, we can say that new methods have enabled a more comprehensive view on diversity, life cycles, and biogeochemical fluxes of pelagic fauna. This would certainly expand the toolset for future investigations. Some guiding questions for our future work are: do high metabolic and reproductive activity during winter imply highly efficient heterotrophic resource utilization? Is animal activity thus decoupled from primary production in large parts of the year? Does this high activity and the year-round presence of predators suggest that the food web in the Central Arctic Ocean may be more productive than previously assumed? This would have implications for future predictions of carbon fluxes and the potential carrying capacity for harvested resources and protected wildlife. Thank you very much for your attention. I am happy to discuss further and answer your questions.